Now we're changing gears. We have Miriam Bravo López. She's a PhD student at the International Laboratory for Human Genome Research at UNAM, Juriquilla campus. And the title of her talk is Surveying Pathogens in the Oral Cavity of Pre-Hispanic and Colonial Individuals from Central Mexico through Paleogenomics. Okay. Wow. That was cool.
Hi everyone, my name is Miriam and I am a PhD student at the UNAM in the International Laboratory for Human Genome Research. And today I'm going to give you a short overview of my PhD research title, Surveying Pathogens in the Oral Cavity of Pre-Hispanic and Colonial Individuals from Central Mexico through Paleogenomics, under the supervision of Maria Ávila Arcos. According to archaeological record, the presence of infection diseases date back to the pre-Hispanic time. Here I show you a map of Mexico with the distribution of the main archaeological sites based on the presence of infection diseases, like treponematosis. I don't think that the color is like
showing. Yeah, something showing.
Like treponematosis and peristitis. Note that this is a scarce record. In contrast, during the colonial period, there are a larger number of historical records. Like, we have the evidence of measles, mumps, smallpox, typhoid fever, cholera, and cocoliscli, which is a deadly disease of unknown cause. So, today it is well known that paleogenomics has offered new insight into the understanding of our coevolutionary history with pathogens. To date, there's only two genome fully reconstructed from Mexican archaeological samples. The first one belongs to Salmonella enterica, which has been recovered from burials, from remains buried in epidemic cemetery in the southern of Mexico. This author suggests that Salmonella enterica to be one of the causative agents of cocoliscli and the cocoliscli outbreak in 1545. The other study entails the identification of two subspecies of Treponema pallidum, and these two genomes were recovered from archaeological remains with characteristic manifestation of congenital treponematosis. So in our study, we wanted to generate data that allow us to genetically characterize pathogens in both periods, pre-Hispanic time and colonial period, and to fill gaps in the understanding of how these pathogens affect the population in Mexico. So to get insight into the past human infection diseases, we analyzed 6 state samples from Toluquilla and Ranas, which is located in the highlands of Querétaro, and 17 samples dated from the colonial period, La Conchita, Coyoacán, which is a rescue excavation located here in Mexico City, and 22 teeth samples from a hospital founded by European missionaries shortly after the conquest in the 16th century. First, the 12 DNA was extracted and converted into Illumina sequencing libraries and was shotgun sequenced. The reads were mapped to the human genome, and the unmapped reads were first analyzed by 2 algorithms to obtain a taxonomy profile. After that, we only concentrate on pathogen matches. We then use PWA to map the reads and to obtain the characteristic ancient patterns of the pathogen of interest. And as we know, the ancient DNA contains a large fraction of environmental DNA, fungal, and other DNA sources. So we use a capture enrichment method to capture the DNA pathogen of interest. Using a biotinely RNA base, and these bases hybridize to the genomic regions of interest, and through magnetic coded strepidine magnetic coded beads, we can select that hybrids, and after washing away the off-target molecules. And we did this kind of method. So what we found is 3 ancient periodontal pathogens, Bartonella forsythia, Parphyromonas gingivalis, and Treponema denticola. These 3 pathogens we found in 1 pre-Hispanic individual and 4 from the colonial period. We have then a really high abundance, and this is clinically potentially significant because these pathogens are the causative agent of periodontal disease today. And these 3 pathogens belong to the so-called red complex, and they destroy the pathogen tissue in a cooperative manner. They are depicted in the top of the pyramid of the pathogenicity of periodontal disease. These 3 pathogens, we could able to see the characteristic ancient pattern of these 3 pathogens, and interestingly, the Bartonella forsythia was found highly abundant compared to the other 2 pathogens. We could able to design custom bank designated baits, selecting 234 genes, informative for phylogenetic analysis and understanding the pathogenicity mechanism. We obtained successfully the capture and treatment of these 300 genes in 5 individuals. Here I show you an example of the reads distribution of 1 individual from the pre-Hispanic period, and the blue one represents 1 colonial individual, obtaining 20.9-fold coverage of the genome. And I would like to mention that previously another study have reconstructed the genome of Bartonella forsythia for dental calculus in Europe, and this study shows the absence of genes related to antibiotic resistance. Specifically, they show the absence of the genes. And our bait design, we included for exactly the same gene, and we also found the absence of the genes in our 5 ancient samples. So the antiquity of the antibiotic resistance remains to be explored in our ancient genomes obtained. And to sum up, we found the presence of these bacteria in both pre-Hispanic and colonial period, and we could able to use this technique, capture enrichment, obtaining to 3.5 to 10% of the reference genome cover, at least 7-fold, and we would like to make further comparative studies between the ancient and the modern strain. And the question that I would like to answer is what's the phylogenetic relation between pre-Hispanic, colonial, and present-day Bartonella forsythia, and also to know the genetic variants related to evolution and pathogenicity of this pathogen in central Mexico. And I would like to thank my advisor and all the people that had contributed to this project.
Thank you, Miriam. The room is open for questions for Miriam.
Great talk, thank you. So I always wonder if with these bacteria that are in the, how are they called? Yeah, so is that merely an introduction, or the change of diet could have influenced their appearance?
Yes, this Bartonella forsythia is also influenced by the type of diet. It's more abundant with a diet based on carbohydrates, carbs. And yes, it's also the lifestyle and other factors that influence the presence of this bacteria. But in the case of periodontal disease, it's more abundant.
Lovely talk. Can't wait to see what you find out in the future in the phylogenetic analysis. But just kind of forecasting, do we know how many mutations we expect to see in comparing an ancient strain to a more modern strain? Like, is there a recombination rate readout or something like this? Like, if you were going to give a prediction, what do you think you would see? Thanks.
Honestly, I don't know. Frankly, I don't know what to expect. And other studies, actually, the other studies that relate the ancient pathogen reconstruction some of them haven't found any change. And so I don't know what to expect. I'm looking forward to do the analysis of all this. Thank you.
Okay. One question here. Miguel.
I was just wondering if you think about the horizontal gene transfer between different species when you think about the species that you identify, and is it even possible, with this type of process that you're using?
Yes, actually this could be. In fact, in antibiotic resistance is very common. So yes, I think that would be interesting to analyze specifically that. Yes, thank you.
Okay. Well, we have few announcements before lunch. But before that, let's give another hand to Miriam.